Hello, welcome everyone for this webinar. My name is Sandeep. I'm the director of Flowthermolab, and today we are going to have a wonderful session by Professor Saptarshi Basu. And before that, I would like to give an introduction about Flowthermolab. If some of you are new to Flowthermolab, so we are an online platform known for computational fluid dynamics, and we have online training on various aspects of CFD, into the fundamentals, into tools, into various specialized topics like turbo machines or multi-phase flow and things like that. And we also do consultancy services for various industries or various companies in mainly from Europe. So that's our background as a company, and and then I would like to introduce about Professor Saptarshi Basu, who is our guest for today. And Professor Saptarshi Basu is a distinguished academic in the field of mechanical engineering, and he's currently serving as the Pratt and Whitney Chair Professor at the Indian Institute of Science, IISC Bangalore. And he earned his PhD from the University of Connecticut in 2007, and he subsequently held an assistant professorship at the University of Central Florida. And in 2010, he joined IISc, where he established a state-of-the-art experimental facility for combustion and fluids research. And you can see, many posts by Professor on LinkedIn on various, fantastic research which is done in Professor's lab. Professor Basu's research focus on multi-phase transport processes with applications ranging from gas turbines and combustion in gas turbines to even surface patterning and many more things. Like, he's an expert in the topics like combustion, instability, optical diagnostic, heat transfer in multi-phase systems, droplet dynamics, and many more in basically into fundamental fluid dynamics. And throughout his career, Professor Basu has received numerous accolades, including the election as the Fellow of Indian National Academy of Engineering, INAE, and also ASME, then Royal Society of Chemistry, Royal Aeronautical Society, and a lot of awards as well. He also was awarded the Swarna Jayanti Fellowship in Engineering Sciences by the Department of Science and Technology, the DST of Government of India in 2014. And Professor did several PhD students, more than 20 PhD students, and he is also editor of several international journals. So he's we have the right expert for us today, to learn from him. And I welcome you, sir, for today's webinar.
Yeah, thank you, Sandeep, for the nice introduction and for the invitation as well. So the idea of today's talk was to give some glimpses into the fluid dynamics research that happens in my lab and a certain thought process that I have that where the research can go, not just in my lab, but elsewhere as well. So these little cartoons that you see around in this particular slide represents, fluid mechanics at different scales. Some of them are large scale, some of them are small scales. So that is exactly what our lab does. So we do not have a bias towards any particular application domain. We basically work in all, so long as the problem is interesting to begin with. Okay. So just a second, let me hide this somehow. Yeah. Okay. So, this particular cartoon basically shows that what are the broad areas of research. So, if you look at the horizontal scale, this basically represents the research that happens from very small scale, which is roughly about a micron and tenths of micron, all the way up to very large scale, which Sandeep was talking about, the gas turbine. Okay, so and all of these are basically snapshots, or basically experimental snapshots of the different types of work that happens in the lab. So I'm going to talk a little bit on this bacterial part, the top left-hand corner, and also a little bit on this, the bacterial droplet impact. So entire the left column, I'm going to do a little bit. And then in this part, coming to the very large scale, I'm going to do a little bit of discussion on this shock droplet interaction. But in between, there are many things, for example, new types of atomizers, injectors, and stuff like that. So So if you look at it, this is more of the same, but this has got an interesting connotation. So if you just see everything by velocity scales, so this part where my cursor is right now represents a low velocity and very small length scale. So what you see are basically bacteria, droplets in microfluidic channels, and these are again bacterial aggregation in an evaporating droplet. So all of these are basically micron-sized events, and their velocities are also very low. Their velocities are also of microns. The middle portion that you see are moderate velocity and small to moderate scales. This one, for example, shows how a vortex interacts with a droplet. This one shows that how a vortex interacts with a porous media. This is the droplet impact on a face mask. That's the kind of face mask that you use during COVID. And then in this particular part, this is the droplets that are generated during an eye procedure. This is actually a human eye that you see. And on the right-hand side, now you can see here, these are very high velocities and large length scales. This is where the gas turbines actually come into the picture. And this one, for example, you can see a flame, very large scale flames. And there will be also, research areas in which the velocity is high, but the length scales are small. And the shock droplet basically fits in that particular category. So you can see that the scales that we cover are basically multi-scale in nature. They cover high velocity, small length scale to small velocity, small length scale to high velocity, high length scale. So all of these things are there in our research. So first and foremost, we do just a quick, understanding of droplets under extreme conditions. And I wrote a few things that why droplets under extreme condition research is important. Because it is relevant to hypersonics, for example, because in hypersonic, you actually have droplets which are injected in a supersonic flow. And also in additive manufacturing, we will see how that happens. And even in spray coating, like solution precursor, plasma spray type of coating, or even in gas turbines, the real high-speed gas turbines. So in all these areas, the droplets undergo very extreme condition. That means it sees very extreme type of flow fields. So these are the two students who are involved. Well, there were more interns and other people as well, but these are the two principal PhD students who are involved, along with collaborator who is Professor Alok Kumar. So the background and motivation, whoever has worked in this area, one can say that, we deal with droplets everywhere, especially in fuel injectors, even in additive manufacturing where you need to produce the metal powder in some way. So that metal powder production also uses the droplet architecture. And at the same time, slightly lower flow velocity applications will include agricultural sprays. These days, with the use of drones and other things, you basically spray insecticides and pesticides and whatnot in agricultural scenarios. You also work in spray coating, for example, high temperature alloys. For example, how do you coat it on a surface? How do you generate a thermal barrier coating on a gas turbine plate? And of course, fuel injectors are routinely used in combustion. The main purpose is that you need to generate droplets which are extremely small in size. So what it involves is basically in a non-dimensional space, you have a high-speed gas which is interacting with a droplet or droplets or a spray, whatever it is. So basically, as we all know, three numbers are important. It's Weber number, Onsorge number, and the Reynolds number. And these are things that most of the people who are working in the area will know. And this actually gives you the domain map or a regime map, which was done by Jerry Fate long time back, where he kind of differentiated the different modes of breakup. And most of you have seen it in some context, one way or the other. But most of the breakup, that was ill understood, and people never really went to high enough Weber number. By high enough Weber number, I mean high-speed gas flow rates. This map actually looks a little empty in that particular way. And of course, these are steady-state maps, so you do not know the dynamics actually. So we look at a few things very quickly, and then we kind of try to rationalize where it can be used. This is the shock droplet interaction of Newtonian liquid. Now here the Newtonian liquid is obviously water. It can be any Newtonian liquid which is like water. So the first case of innovation that has come out of this particular work is that we have developed a miniature shock tube. So normally, as you know, shock tubes are very large. They occupy a lot of area, and it's very hard to actually do experiments with shock tube due to multiple reasons. So here we actually did an innovative thing. We coupled the shock tube with a levitated droplet, and this provides the kind of fidelity that you need to see what is going on with the droplet when it interacts with the shock. And on the right-hand side, you can see that the Mach numbers that we can go to, so it's roughly of the order of 2. But the important part is the Weber number, where we go more than 10 to the power of 4, actually pushing much higher than that. Technically, there is no limit. We can go actually up to Mach number 4 as well. All right. So what people did not see probably, and this is what we see, in this unique set of experiments. So what do you expect when you actually have a shock interacting with a droplet? First, you see the shock. You can see this is the shock front. It interacts with the droplet. You see some reflected shocks, and we will explain what those things are. Nothing happens to the droplet per se for the first initial time period. The shock seems to have come, it seems to have gone. But then it is the flow that is generated behind the shock, which actually then leads to the spectacular breakup modes, which we call, we can go into more details if somebody is interested later. This is called Rayleigh-Taylor piercing, and this is called shear-induced breakup. So these are the kind of modes that you get. So if you look at on the right-hand side, you can see a small video that what happens when the shock actually interacts. So you can see all these stages actually happens. You see a whole gamut of dynamics like transmitted wave, then reflected wave, then focusing, and Mach-Mach collision, and then a recirculation zone formation. And because the recirculation zone forms, there is a pressure differential that is created across the fore and aft of the droplet, which leads to bulging and which leads to atomization. So this is a fundamental insight that you can get using very high quality diagnostics. And what happens, that this is, for example, happens at 1300 plus, the Weber number 1300 plus. You can readily see that there are formations of these waves along the droplet surface, which in the fluid dynamics lingo, it is called the Kelvin-Helmholtz instability. And then after the instability starts, you have mayhem after a little later, and you get all this kind of crazy breakup characteristics. Okay. So, and I will play this video, and I will play this video also. You can see that as you go on increasing the Weber number, things gets a lot, it's really hell breaking loose, so to say. Okay, but the inception point, if you look at it carefully, are the Kelvin-Helmholtz waves that actually happens. And this, if you look at it in a very zoomed fashion, you can see the waves quite clearly. The liquid sheet that is bulging out. So basically, there is, as the droplet deforms, there is a liquid replenishment that tries to go towards the edge, and that leads to the formation of ligaments and and ultimately atomization. So this is what we call the atomization of the peripheral sheet. Okay, this is the shear-induced breakup mode. In addition to this, if you look at it from the top, you will have, because as you know that this is a droplet which is basically pushing through a lighter fluid essentially. So what you have is what is called the Rayleigh-Taylor modulation or instabilities. So this actually sits on the top of the Kelvin-Helmholtz waves. So the Kelvin-Helmholtz waves are modulated, or the Artin waves actually modulate the Kelvin-Helmholtz wave. And if you look at it from the top, it looks like almost this sun corona type of thing, where things are coming out from the top. These are kind of insights that you would normally not get, okay, unless you probe very deep. So we were the first, basically, to propose that which of the mode will become dominant. We were the first to propose this criteria, which tells you that where the shear stripping mode will be more dominant than the Rayleigh-Taylor piercing kind of a mode. Okay, it's a long story. We'll talk about the physics a little later if somebody is interested. But basically, this was a unique finding because we could say when the switching actually happens, and this is a function of the droplet size as well. So so to cut a long story short, larger droplets, you have more propensity to have the SIE kind of a breakup mode than smaller droplets. So the idea is that the larger droplets break up to the SIE mode, and then the smaller droplets suffer the Rayleigh-Taylor piercing. So it's a beautiful cascade. If you look at it, the droplet kind of self-selects itself in that very unique way. So that is the story with the Newtonian fluid. Now, coming to the polymeric fluid, we'll see where the polymeric fluid is very important. This is, for example, all of you, after having recovered from the bout of COVID, right? You know that this is what comes out of your mouth when you actually sneeze. So this is very polymeric, as polymeric as it goes. This is, for example, a pesticide spray that actually happens. Okay, so this can be also done by a drone, by the way. So polymeric droplets are there everywhere. And now, even in the energy sector, with the advent of slurry and emulsified fuels, for example, fuels which has got extenders in it. So those kind of fuels also are polymeric in nature. They are basically complex fluids. And also, that, sprays of this kind of, pharmaceutical chemicals, fruit pulps, whatnot, you can actually have polymeric droplet atomization. So in addition to the numbers, Onsorge number, Weber number, and of course, Reynolds number, this is the Mach number. So you also have something which is called the elasticity number, which tells you that it's kind of a relaxation time because it's a polymer. So basically, it has a relaxation time associated with it. So how does this actually change the breakup morphology? Whatever we have seen earlier, does it apply over here also or not? Does the droplets have a universal atomization framework? That is the whole point. You can look at a few observations. These are actually spectacular images, as you can see. This is what I call the jellyfish type pattern. Okay, so as you can see that as you go on increasing the polymer concentration, so this PEO, that whatever you see, that is basically increasing. So what happens over here, and as a result of that, the elasticity number increases. It increases by actually 6 orders. So what you get from these tiny ligament type things to a very, to a sheet which is crumbly but doesn't undergo atomization. So as you go on increasing the elasticity number, atomization is kind of suppressed, even at very high Weber number. This is Weber number more than 1,000. And these are all very comparable Weber numbers, as you can see. You can never match the Weber number exactly because, experiments are experiments. CFD guys will have a better field day with that. So what we did was that we again did the kind of, regime mapping over here, as we saw that there was a deformation. And depending on what is the Weber number, you still see the advent of the same Kelvin-Helmholtz wave and the same KH and RT wave modulation on the top of it. So up to this particular point, before it goes to what we call the stage 3, the dynamics is universal. You still get the same mechanism, the same KH and the same RT waves. But the ultimate topography, of course, is very dependent on the elasticity number. So what we have unearthed over here is something which is really interesting. That means the inception before it goes to the topology phase, okay, the mechanisms remain the same. In other words, the forces that are responsible are still the same. It is only at the end stage that you will have the topographies which is determined by the elasticity. So like, for example, you have a viscosity which is a time delayed player in your breakup dynamics. Elasticity almost plays a very similar role. I mean, it's almost you can draw a parallel with respect to your viscosity. So this gives the map that if you go on increasing the Weber number for one particular elasticity number, things gets very messy. It's supposed to happen because we are going down to almost 10,000. And if you go on increasing the elasticity number for a given high Weber number, you can see that the breakup becomes, it looks shabby, it is still catastrophic, but it tends to get a lot more well-behaved, so to say. All right. So the presence of this elasticity, okay, it transfers it to a more stabilizing kind of a domain. But the inception phase, that is up to stage 2, it is still KH and RT. It is only at this stage that it acts as a stabilizing stabilizing anything, so to say. So that was fantastic. And all of this can be put now in a time map. So up to this particular point, everything is the same. And then things actually bifurcate. This kind of branches out after that. Okay. So the breakup modes are still governed by the Weber number, while the breakup morphologies actually depend on the elasticity number. So that is fantastic. I mean, so we do have kind of a semblance of a universal framework in this kind of high-speed applications. So this is a cover art which came out in JFM. So now that we did polymeric and Newtonian, let's look something at liquid metal, because you need liquid metal for metal powder production. You need it for thermal sprays. You need it for additive manufacturing and whatnot. So for producing powder, you need liquid metal. And for coating surfaces, also you require liquid metal. So liquid metal is there. So this was the same miniature shock tube facility with a little bit of a gizmo added to it so that the metal does not oxidize, basically. That's the whole point, to prevent. So it's done in an inert atmosphere. All right. So this is the metal atomization phenomena that you see. So you can see that when the metal actually atomizes, it comes out as flakes because of that oxide formation. It's like almost like an enhancement of its surface tension, so that it comes out as this kind of flakes. When it is not oxidized, it behaves very similar to that of water. Okay, if you compare this and this, well, if I don't tell you that one is water and air, and the other one is a liquid metal, you won't even recognize it. So and remember, there is a lot of difference between their properties as well. Okay, but the idea is this picture itself should suggest that the universal framework works, because you cannot make out the difference, even though one is a liquid metal and the other is water. So this kind of says that, okay, we have been able to nail the atomization modes. We were also able to nail the topography part of it. What still is missing from this particular point is this. So our study now can enable you to develop new class of injectors. So you can use a shock enhanced injector, which will be very novel, and it can be used in hypersonics, because in hypersonics you really have very low residence time. So therefore you need to complete everything within a very short time span. This is actually a closed couple atomization, which is actually used for generating metal powder. So improvements of this can be done. So things like that. But the most important part that I'm going to harp over here is the last point. That is that with the advent of this machine learning based models, essentially data science, what you can do, what we were unable to do at this particular point, is that if you look at the breakup, the real final morphologies, it is impossible to predict and even do anything realistic with it. Okay, but this particular morphology is actually what determines the droplet size distribution at the end of the day. So if you want the droplet size distribution, okay, you really need to have the machine learning, which can predict this droplet breakup topologies and atomization characteristics. Because industry people, they would want you to predict the droplet sizes rather than all this, great insights into how the breakup happens. They would really know what is the outcome of the breakup. So that is the most important part. And at the same time, CFD solvers, you can actually provide very high quality, benchmark quality data for these kind of applications. So it has got two very distinct modes. One is very fundamental, where you actually probe what is going on. And the other one is very applied, where you design new injectors and you develop all these crazy machine learning type models to basically help you design injectors faster. In fact, that's what the AI ML is supposed to do. So this particular small segment, now you saw that this breakup was catastrophic. I mean, it generated droplet within a very short time span, very high Weber number events. So if you really want to measure the droplet size, because that is your ultimate aim, and you also want to measure the droplet size distribution and the dispersion and stuff like that, how do you actually do it? Your normal PDPA technique is not going to work, just because of the fact that PDPA requires a lot of time and requires a lot of samples. Your shadowgraphy is not going to work because things go out of focus very easily. You need a very small depth of field. So what we use is a deep learning technique, which is a depth from defocus technique, where you don't really need things to be in focus at all. And so this is actually a shock droplet interaction that is actually going on. And these are basically the number distribution and the diameter distribution that you get as a result of that. And not only that, you can do it at very fast. Well, if you can see, this is done at Weber number of 4,000, which is very high, pretty much unprecedented. And we use a two-camera technique. And well, the technique we can discuss later if somebody is interested. But the idea is that you can use this framework now to measure droplet size distributions at different points in time. Okay, and that is possible because it is impossible to do it in any other way unless you do it in an X-ray or something like that, which is very expensive and does not exist. You can actually do it, and all you need is a high-speed camera and a laser, and even an LED source is going to be just fine. Okay, so this is all you need. So your speed of the high-speed camera is actually determines how fast you can do all these measurements. All right. And not only that, so this technique was very good for high-speed applications. And we also showed that this is applicable for very low speed and other types of applications like pollen grain, measuring the pollen grain. This is, for example, surface bubble bursting and the droplets that you generate. This is, for example, maybe like an inhaler spray that you take. It can measure that. It can measure glass beads. It can measure many kind of stuff. And it enables you to have a lot of applications, right from high speed all the way up to low speed with very basic instrumentation. It's much, much cheaper than PDP or any other technique that normally people would use. You obviously would still need optical access to do this, but that is what it is, and it can be done. So now that we shown that, okay, you have a universal framework, you can still need machine learning, but with experimentally, you can still measure the droplet size. So maybe that droplet size information and the universal breakup phenomena and the topology using the AI/ML can all be merged into one, and we can get a framework in which we can predict in this kind of catastrophic events, be it a combustion chamber, be it an additive manufacturing chamber, or be it thermal spray applications, we should be able to predict the outcome. That is how this droplet dispersion and size actually happens. So now that we have done this, these were all non-reacting to begin with. We did it also for shock and reacting flow configurations. And these are the two students are involved whose names are right over there. So these are two different students, not the same students that we did the previous work, okay, because this is a lot of work. Okay, let me tell you that. So I need not say any more. We have auto-ignition of fuel in supersonic flows. We have shock flame interactions in supersonic flows. And most of these flows actually use both gaseous fuel as well as multi-phase systems. So we did both. Okay, so we did, these studies for both types of stuff. And this is also interesting because nobody had really done it in this particular way. So this is the shock droplet interaction. So this is the same shock tube setup. So this is the beauty. You generate the setup once. Now you can use it for many things. And you need to be a little bit innovative in this particular category. So what happens over here is basically you have a flame which interacts with a shock, a droplet flame which interacts with the shock. And you see these beautiful things. You first have a flame shock interaction, and these videos actually show that. Then you actually have a flame-induced flow interactions, which is exactly the way that the cold flow and the droplet behaves. And then you have a droplet flow interaction, which leads to atomization. Like a three-part event, which repeats itself. So, and all happens, as you can see, the time scale is really, really short. Okay, so here the droplet breakup that we showed, before that, a lot of things happens, a lot of action happens before you go to the actual droplet breakup phase. This is a complicated diagram, so I won't bore you with that. So these are different flame transitions that we actually show when the flame interacts with the shock. Why this is important? Because in an actual combustor, when the flame behaves like this, it can actually have reignition, it can have extinction events that can actually happen. And once you do this, you basically lose propulsion. If you're using it in a hypersonic case, you're basically losing propulsion. And you can also have combustion instabilities that actually can happen in this hypersonics. Because remember, in hypersonics, the residence time is rather short. Everything has to happen very, very quickly. So these are kind of a regime map, equivalent regime map which shows that how this droplet flames actually behave. So that was the droplet flame. This is the jet flame. So this is a gaseous fuel right now, which interacts with the blast. Same setup. That's the beauty part. Forget about all these other variations. You can see essentially it is the same thing, except now that there is no droplet phase anymore. So you have a flame shock, which is blast here, interaction, and then you have a flame-induced flow interaction. And the flame shows all these spectacular characteristics. You can see there is vortex roll-up, there is vortex shedding, and all these things. So actually, even if your hypersonics now uses a gaseous fuel, you should have characteristics like this that happens. Okay. So, and these are extremely high-speed events. And this is one, some of the, attachment and extinction events that we have discussed. And there has been a lot of work that has gone on that how the baroclidic torque actually leads to vortex roll-up events and how the flame shedding happens, how the flame reattachment happens. All of these things are actually, actually done over here. So this is a very extensive set of work that we have done. Okay. So why these studies are important? Sorry. Once again, this studies has a fundamental connotation in which it is interaction of flames with nonlinear pressure waves. So if you look at it that way, if you solve this problem, you solve combustion instabilities, you can get further insights into flame extinction events. This is the same thing is applicable in hypersonic combustors. So again, the machine learning part comes into the picture because you have these zillions of flame response behaviors, and some of them actually lead to instabilities. So you can use machine learning to develop a framework in which you can predict instabilities by noting certain types of flame responses. It would be amazing. And also new technologies like in detonation-based engines and even in rotating detonation engines, you can actually use this kind of studies to develop pre-detonators, where you use this shock flame interaction to achieve this deflagration to detonation transition. So these are very exciting pieces of work, which are again fundamental, and at the same time, they have got a lot of applications. Depends on where you want to park yourself. You can park yourself in both. That is not a problem. So all these things that I showed up till now is high speed, and well, they are kind of small scale still. Okay. So now we go to an area where everything is small scale. That means the flow is small, the size is small, everything is small. Essentially, we go to something like bacteria and stuff like that. Now, this is exciting because some of you may see, okay, people who work in combustion, people who work in shock normally don't work in, bacterial droplets, so to say. But we are a group, as a group, we are a little bit different. We think that these are all droplets at the same time. They evaporate, they exhibit flow. So how does it matter whether there is a blast going on or whether there is a simple evaporation problem? So if you look at the small scale hydrodynamics part, I'm going to just show once again brief snippets. So we are going to cover, after COVID, there has been a lot of things that is happening in the air that, all these pathogens, how do they respond and how do they become more virulent, whether that virulence has something to do with the mechanical stresses or not. So a bacteria or a pathogen might become virulent in a varieties of ways. Like we hear the Delta wave of COVID. Of course, that was a virus, but we have always seen bacteria which have morphed, which have kind of mutated and have become extremely resistant to antibiotics, for example. That's a problem that we know. So is it just because of the antibiotic, insult that we do to the bacteria? But also, does it have anything to do with the stresses that the bacteria faces in real life? So like normal biologist, you would do it in a petri dish, you would do it in a lab setting, which is not a realistic atmosphere. So we'll try to see that how these bacteria respond when you place it in realistic conditions. And what are those realistic conditions? The bacteria can be contained in a droplet and it may be sitting on a surface and it may be drying, right? For example, in pneumonia, it can dry on a surface. That the bacteria may be in a droplet and it may be actually floating. So it is like an aerosol. Then this bacteria, when we cough or if the bacteria is contained in water, these droplets comes and impinges on the surface. So there is an impact that actually goes on. And impact problems have been studied by many people over the years. And the last one is that what about our own body, these bacteria and other things, when it actually circulates through our blood, through these small capillaries and stuff like that, they undergo stress because like any other particle, they should feel some kind of a shear stress. So what happens to them? How do they respond to that? And sometimes the stress can be quite high. So that is one part of the argument. The second part is now using that understanding, can we make this point of care kind of a diagnostic framework? Now, droplet microfluidics has been already there. What we propose, and I'm not going to show it over here, is something called a lab on a drop kind of a framework. Then also the droplet drying type of thing, like blood, urine, and stuff like that. Can they be used as a disease marker or a disease diagnostic thing? And you already have the framework of organ on a chip, in which you replicate the human capillaries into a chip and try to see that, what kind of, you can test drugs, you can test tissue samples, you can check bacteria as well. Okay. So there is a whole lot of things that are going on right in this particular slide. Okay. And again, these are the students. One of them is a postdoc actually. And this is the work done with a microbiologist in IISC. Okay. So, idea is that bacteria are pretty smart people. If you consider them to be smart people. So when they see some kind of a stressor, they change their physiology. Okay, how they change their physiology? We human beings take a lot of time to evolve. But the bacteria are single cells, so they can quickly evolve just like that. Okay, so our question is very simple. What happens to the bacteria under evaporative stress, under impact stress, and under shear stress? Very simple. Three things. Okay, so out of this, you can see the evaporative stress is the slowest because you slowly desiccate the bacteria out. Impact stress is short, is high, but it is short-lived. And motion within a microchannel is a continuous stress, okay, which is somewhere in the middle of these two in terms of magnitude. Okay, so let's see what happens. So this is the impact stress. Okay, so the bacteria is impinged, bacteria-laden droplet impinges on a surface, okay, and then it dries. So what happens is that it's a mixture of impact stress first, and then you have the drying, the normal drying of the bacteria. So what you see sessile over here is basically the droplet just drying, and 5, 7, and 10 are basically the kind of velocities with which the bacteria is impinging on the surface, right? So that is what happens. So idea is, and to cut a very long story, very short, if you do this experiment and if you do it properly, you will find that, look at this last bit of graph over here. Bacteria under no stress is under lab environment. So it shows a certain amount of virulence. Virulence means its infectivity capability. Let's take it like a very fuzzy term, but that's what it is. And then the middle one, which you see over here, is just drying. That means you place the droplet on the surface, allow it to dry. So you can see that it increases. And the last part is desiccation and impact. So that means it first impacts and then it dries. Here you see that as you go on, doing this, the bacterial virulence actually goes up, but the viability comes down. Another way of telling this is that, say you start with 100 bacteria, okay? When you have an impact and desiccation, only say 10 bacteria survive. But this 10 bacteria shows probably 2x virulence. So they act like 20 bacteria, if I have to put it very, very quickly. This is what happens. And so you can imagine, so the environmental factor like desiccation and impact has made the bacteria more virulent. So it's like essentially survival of the fittest in a it's a microcosm of the survival of the fittest, right? That's what it is. Essentially, that's what it is. If I have to put it like that, so that is what it is. Rest are all in the details, as you can see. Not going to go through the numbers over here, So basically, we tried it with different, physiologically relevant medium, that is cough and all these things, gastrofluids and stuff like that, where the bacteria normally is found. Okay, so it's not just found in water, it's found in physiologically relevant mediums. And these are more characterizations that we did. So the important part is that as we go on increasing the stress on the bacteria, that means the impact stress, which is a function of the Weber number. Higher Weber number means you basically splat the droplet with a higher velocity. That's all that there is. Now you can see that as we go on increasing the Weber number, the PHOP genes were seen to be upregulated by the bacteria in order to encounter this kind of an impact stress. And this is there. This is right. The evidence is right in front of you. So this was great, so to say. And we did the same study on liver and spleen. That is, this was animal infection studies. And we see that there is a higher proliferation, which indicates higher virulence, just because of the impact of the desiccation stress. Exactly what I told you over here. Okay. So this I'm going to just skip for a while. Okay. So let's go to the stress on the bacteria inside microchannels. Now, why this is also important? Because, organ on a chip that you see right over there, this is a complex network. And, so what happens is that, any bloodstream infection, what you see over here, okay, actually this bacteria and other things passes through the blood, is a universal carrier. It's a circulatory medium, so to say, that has got its pros and cons. So we kind of investigated it using an unstressed bacterial sample and then basically doing a pulsation, right, to stress it out and see what stress and for what duration of stress we basically get what kind of result. So again, the same story is valid. The unstressed bacteria are lower in virulence. The stressed bacteria are higher in virulence. Okay, and that again happens due to the gene and the gene upregulation. Okay. So these are more details. Once again, if anybody is interested, feel free to write to me. So the channels are intentionally made this way because our human capillaries can be complicated. Okay, it can have more complications than this, but honestly speaking, doesn't really matter that much because it's a more fundamental way of looking at things. So you varied shear. These are very realistic shear rates, what you are going to have in your human body, for example. And the stressing time is from 2 to 9 minutes, which is also kind of realistic. So using that, and then we study what happens to the bacteria. So intracellular proliferation means that how the bacteria kind of infect the macrophages. So here you can see that with the increase in stressing time and stress values, the proliferation basically doubles. In other way of saying that the virulence actually doubles. Not a very long story, very short. Okay. The other interesting part that you see that unstressed bacteria are very round and it kind of looks like peanuts, but it's not. Smooth edges. But the stressed bacteria actually develop these dent-like buccal structures, okay, which happens because of the stressing, because all other parameters are basically the same. And this stressing actually leads to a change in their aspect ratio as much as 10%. Okay. So you have seen the channels. Now, if you also look at the bacteria as it sits on any substrates without any initial force, okay, this is important as well because you used to clean your Amazon packages during the COVID time. You used to sanitize it. You used to wash your hands when you touched the handle of a bus or touched a coffee mug and stuff like that. So the idea is that it is very relevant to see that in those dried configurations, which is on substrates, which are basically fomites, how does the bacteria actually behave? So as you can see that if you look at this picture over here, the bacteria mostly accumulates at the edge. This is like a typical porphyrin type of a pattern. And you see this interlocking network inside it. This happens due to thin film instability at the center of the bacterial deposits. Now, at the edge, there is a lot of squeezing of the bacteria. This is called the rush hour effect, that where the bacteria are basically squeezed, and because they are deformable, I mean, rigid spheres, there is a certain packing fraction that you can achieve. But in this case, you can actually stack them because of the deformation. You can deform them and you can pack more. So that's how the squeezing actually happens. So these are actually the squeezing images. So if you look at now the SEM images, you see this side and this side looks a lot different. This is the rush hour effect, and this is the actual nice packing. Almost looks like a cemented edge. So this outer edge looks like this, the inner edge looks like this. All the bacteria develop this very strong buckled site. This happens due to the desiccation in this particular case, because it loses water. As a result of that, the capsule actually inverts in dimension. So, okay, we are not going to go what Voronoi tessellation is. This is once again, mapping out the patterns in the deposit. And the bacterial elasticity also varies quite a bit, interestingly, with the location of the deposit. So the outer edge bacteria is very highly elastic compared to the bacteria at the center. And the Voronoi area also varies due to this squeezing and this random packing during the rush hour. Random packing is something that is not new. It has been there for quite some time. Again, if you do the infectivity analysis on this macrophages, you see the peripheral bacteria survive much better than the bacteria at the center because they have some kind of a coral. Okay, so they can actually pull the food together, so to say, because it's kind of like a colony which has been brought in by this squeezing action. And the peripheral bacteria are highly infectious at the same time. So if you have a precipitate and if you touch the edge, the chances of you getting infected is more than if you touch the center, which is kind of who would have thought about this spatiotemporal map, right? So you do the same thing now with the substrate as well as with the contact-free droplet, which is essentially the aerosol, as we call it. So, like for example, when you cough, it can settle on the substrate. It can also remain in the air. So, which one is more infective, so to say? Which one actually shows more infectivity? So, this is a typical evaporation curve. I'm not going to go into the details of this. So, in the levitated mode means when the droplet is contact-free, the bacteria accumulates at the interface of the droplet. And in the sessile mode, it is at the droplet contact line. That is where the bacteria happens. Okay. Now, the bacteria has got a higher viability. That means the survival rate is higher in the levitated precipitate. And also, it shows more infectivity in the levitated precipitate. So, if you are going to inhale aerosol, your chances of getting an infection is much more than if you are going to have a sessile droplet, that is Fomite. So that's actually the infection. This has nothing to do with the bacterial type does play a role because you can see all kinds of bacteria. You can see pneumonia, you can see, Klebsiella, and all these other bacteria are over there. Right? So the idea over here is very simple, that it is crucial. What I'm trying to emphasize is that the dynamic environment of the bacteria is generally not considered in the batch culture that you do in the lab. That is very important because as we rightly showed, that it can have a tremendous effect on the viability and the infection because the bacteria senses it, then it upregulates a certain gene, and therefore it can actually become more virulent. And if you want to model epidemics like pandemic or diseases like tuberculosis, you have to understand what is the effect of these evaporative and impact stresses, because this is real. Okay? And also, if you want to model bloodstream infection like UTI, for example, you have to understand what is the shear stress that is produced inside the bacteria. Or if you want to consider a biofilm that forms inside your body. So, to develop strategies to mitigate the spread of the disease is not just enough to just look at the biology in the lab environment. You have to couple the fluid mechanics and the bacterial physiology and develop this new class of studies where you understand that how the bacteria actually behaves, okay? Or how the bacterial evolution actually behaves, which is tightly coupled to its transmission. So we'll spend a few more minutes on the point of care diagnostic part of it. So, and this I'm not going to go into detail. So on the left-hand side, you can see a blood droplet, which has got bacteria. And on the right-hand side, you actually have urine, which contains E. coli. and the subject is a female and a male. So I don't have to tell you much. The urine without the bacteria shows only small crystals. The urine with the bacteria shows a lot of crystals. So the presence of certain biomarkers and its interaction with the bacteria manifests itself in the dried pattern of the physiological fluid. In naked eye, you can actually make that out. So how about you if you use this physics-informed neural network type of models to generate this very low cost, very preliminary diagnostic tools. And we are not saying that this is the absolute gold standard marker. For that, you still need to do you need to do culture studies on this. But if you are going to use this low cost POC data, especially in the poor sectors of the country, where you need a quick turnaround time to see whether a person is infected with E. coli, that is UTI infection, or somebody has got a bacteria in its blood. If you can do that, at least at a very crude level, you have done your primary sampling. So that could actually save a lot of human lives, okay, before you actually go to the next stage and try to, harp on that the gold standard diagnostics are the way to go. They are the way to go, but these can provide very important insights and very quick turnaround time as well. So I'm going to just end this talk with a little bit of work that we did during the COVID response. And this has got a different set of, again, students, people, collaborators, whatever you call it. So the COVID response is something that we started working on during the COVID time, obviously. And now, anybody here also, it's a little bit of innovation. Now, COVID transmission, people normally think it's pandemic models. There are epidemiologists who do all these pandemic models, but the epidemiologists are not fluid mechanics people. So they don't really have an idea of what is going on at the droplet scale. So what we use is basically multi-parameter regression fits, basically to get the inputs to their macro scale, pandemic model by which they can predict what will be the infection rate and stuff like that. We decided to do something different. We decided to combine this droplet physics-based small-scale model and a chemical kinetics-based pandemic model and merge them together so that you remove the, the hand waving or the ad hoc assumptions as much as possible. Okay. So the idea is very simple. That when a population interacts, here is a population, here is another population, you need to predict how fast the disease spread. This is a very simple question which can save life, because that's where you can prepare your hospitals, you can prepare your oxygen cylinders, and in the Delta wave, this was really fast. So therefore, we could not catch up. So the idea is that in your pandemic model, the rate of infection, the rate, which is I or K, that depends on a lot of parameters, and the droplet is one of the parameters, because the droplet evaporation transmission is what actually led to the spread of COVID in the first place. It was respiratory. It was mostly through respiratory fluids. So as we talked, as we coughed, as we sneezed, this was the one that actually was there. So we did some fun stuff. We modeled your cough. There's a violent coughing. We modeled your cough. Then we modeled the aerodynamics of the droplet in that cup, okay? And then we had a 0th order heat and mass transfer model. Why 0th order? You can add more fidelity. We ourselves have added more fidelity by going to very intricate three-dimensional droplet models. But the idea over here was to get a quick fix so that it can feed to a large-scale pandemic model without where the computation time is kept to a bare minimum. Right? So input included everything: the evaporation rate, the droplet size reduction, the presence of the non-dissolved, non-volatile components like salt. Your sputum has got actually salt and mucin, and the crystallization, because these are salts, so they will crystallize at some particular point of time, and energy balance at the end. So using this, we parked it with experiments. These were actual experiments that were done. The match is pretty good, if you want to say it like that. So, and we, redefined the Wells curve, Wells curve basically says that the smaller droplets evaporate quickly and remain airborne, which is kind of logical. Larger droplets settle quickly because of gravity, because of the Stokes settling velocity. So you can understand, smaller the droplets, more dangerous it is because it will float around. But larger droplets will, be deposited on the ground around you. Okay, so these were the modeling results. And here, if you forget about everything else, just look at the 2 points A and B, for example, right? So what it does, what I'm trying to say is that the droplet lifetime, that means your half droplet has a much higher lifetime when the humidity is high and the temperature is low. And it's the opposite when the temperature is high and the humidity is low. That means if you are in Rajasthan, for example, your droplet lifetime will be very short. And if you are in a seaside, okay, in a cold New York winter, for example, your droplet lifetime almost increases by 1 order. Now, the interesting part is that if the droplet lifetime actually increases by 1 order, this also means that the convective transport of the droplet is also much, much more. That means when you actually cough, your droplets can travel much larger distances. So it is much more than 6 feet, as you can see over here. Okay, it's much, much more than 6 feet. It's more like 1.34 meters, so to say. So as a result of that, what you see is that 6 feet distance, whatever, in the COVID guidelines it was prescribed, is just a number that people pulled out. I mean, I don't think there was much of a rationale with that. Much better rationale comes from just doing this very simple exercise. And we are not moving far away from our field. There's still the droplet evaporation, vaporization, and dispersion that we normally do in a spray, in a gas turbine, in a spray dryer. Okay. So these are basically the insights from the model. Now what we do, now you have this small scale models now. Now you develop a pandemic large scale model, which is basically, you see, people who have done in combustion, they know that there's something called chemical kinetics, which is basically 2 species, how they react and form a 3rd species. How fast does this happen? So if you have a model which is called SEIRD model, here it means S means susceptible. It interacts with an infected people. It becomes an exposed people and an infected people. Exposed people becomes infected after a certain amount of time, and the infected people either recovers or dies. Now, in the COVID, it was about 3% initially. That's why the rates are like that, right? Now, this K1 that you see, how the infected people makes people susceptible or make people exposed, this K has to be much, much greater than these 2 Ks for the disease to progress. Otherwise, the disease will fall flat. Okay. In some cases, in some viral infections, it really happens that way because it does not spread, it's locally arrested. All right. So the K is a prime mover over here, right? It's much fast. So where does this K come from? It comes from your droplet model. So you fit the droplet model into this K, and therefore you get this very nice, pandemic model. This becomes ultimately an eigenvalue problem. So you solve this particular problem by coupling the K, and all these parameters that you see comes from your droplet model, fit it into K, and this is the large scale model that you solve, and you get your final outcome. And what are those outcomes? This is the outcome. This is the susceptible, exposed, infected, and recovered curve that you see for a certain temperature and relative humidity. And this is the probability of infection. Now, what it shows really, just to cut a very long story short, D means droplet. This blue color means nuclei. Nuclei is basically the aerosols. So what happens is that initially, the direct mode of infection is very high. That means I cough, the person in my vicinity will get infected first. But then I cough and say I leave the room, but this nuclei remains because it takes some time for the nuclei to form. This nuclei hovers around, and a person coming much later can get infected. So the aerosol mode of infection, which is this blue curve, actually picks up as this direct mode of infection drops. But the idea over here is that while the direct mode of infection probability is very high, this probability is way lower. So that means why we survived during the COVID. Because if it was the case that the aerosol mode of infection was as effective as the direct droplet mode of infection, half the population would have been gone. Okay, because you cough and you sneeze, you leave the nuclei in the room, somebody entering after a day will get infected in the same way, right? So initially the risk from the droplet is higher. Later, the risk from the nuclei is longer. So that means there is a short-term time scale and then there is a long time scale. So this is interesting. This is how you get realistic answers by combining what we call the droplet model with a pandemic model in the first place. Okay. We also did some work on mask. This was also part of the day. And here the most interesting part is that most people work on mask in which they, look at leakage. So when you wear a mask, things gets leaked from the sides, right? And but here what we did was that what happens after wearing a mask if you sneeze or cough? Okay, so this is your mouth side. Okay, so you cough, the droplet actually impinges on the mask, and because the mask has got holes, because it's porous, the droplet extrudes out, okay, and becomes smaller droplets. So what happens is that you started with a very large droplet, you generated a lot of small droplets. Now you might ask, why is that relevant? Because large droplet would have settled in your vicinity. By doing this extrusion business, you have generated a lot of aerosols. And just now in the previous slide, we showed that aerosol is bad, very bad, because it floats around in the air and it can cause viral infection. So that's why improper masking was very, very bad. So that's why you needed the 3-layer, not just because of the leakage, but because of this. Okay. And okay, this is an extremely nice video that if you cough and you generate multiple droplets, you see, I did not talk much, but you can see the things looks pretty scary, so to say. Large droplets on the top, small droplets coming out. So your mouth is on this side, and this is the side in which the other person is actually sitting. Okay, so the risk from aerosols actually goes high, and this is a very complicated one. We did a lot of fundamental stuff also on this. We did a lot of ancillary stuffs also on this. So as you can see, once again, this is an impingement problem. This is an atomization problem in which we specialize. We just have applied it to a slightly different domain, but the physics essentially still remains the same. So we are able to do all this. This is the team. These are my super duper students. Some of them have graduated. Some of them are still there. Some of their pictures may not be here. So sorry about that. And these are the funding agencies who have contributed towards what I call my journey in intellectual pursuit. And thank you. So that's
Thank
it.
you. Thank you very much, sir, for the wonderful talk. From hypersonics to additive manufacturing, machine learning, then marrying with microbiology, then pandemic modeling, mathematics, and it's a wide range of application of fluid dynamics, and it's really insightful. I hope all the participants they benefited out of it. Most of them are from the CFD background, but to know the real world experimental side, it's really insightful, and very thankful for you for the great presentation, sir. So maybe we can take few questions. Those who have questions, you can also put it in the chat, but they have already sent a few questions during the registration. So
Yes.
maybe if few of them like very generic, which may be useful for others, we can
So I think one of the question was, I guess, that whether, where the research in fluid mechanics is going to go.
Yes.
What is the role of data science and AI ML? I think I already presented in the presentation.
Yes.
It's very clear that, with the advent of this, basically it is some version of a reduced order model, if you think about it that way, except that if you use physics informed neural network type of stuff. So you are basically incorporating a lot of this, physical understanding into something which is like a multi-parameter kind of a curve fit, so to say, if I have to put it that way. But that has got its own usefulness because, this is the way, because some of the things you cannot do mathematically or experimentally, and you cannot have a theoretical basis. You can only provide some, consistency. You can provide a framework. And that's where I think this would be a very important thing, especially in that field of point of care diagnostics, where I mentioned physics informed neural network, as well as in these droplets in large scale applications. somebody who is working on combustion instability can also use the same thing for instability prediction. I think there is a lot of work that is already happening in that particular area. So yes, the field is fast moving and one needs to adapt and one needs to see this as an opportunity, but use it kind of, responsibly. So it should not be used just as a black box completely. We should have an understanding of what is going on. So I think that was one question. The other question was that on these dried patterns, that where the research can go. It's very attractive because, for poor countries, not just, India is no longer poor. It's like a lower middle, I think, income country, but say African countries, for example, and where, the infections, the bacterial and other infection probability is quite high. There to have some kind of a sorting tool, which allows people without the intervention of doctors and even qualified nurse. You just have this like an Anganwadi type of, people or social workers. If they can do that, and we can somehow couple this with an AI type of a thing, that everything is in the cloud. So there is potential where you can actually do a rapid screening of a very large population. Your accuracy may be somewhere in the 60s and the 70%, I should say, maybe 75 if we push it higher. But that's okay, because at least you would be able to save 75% of the life. So I think that is a very big bonus in utilizing this. And this doesn't get any, it doesn't get any cheaper than this. We are not even using a microfluidic framework. So there is the fabrication cost is rather low. But once again, there's a long way. You have to do a lot of clinical trials. You have to get to that particular stage. What I showed over here with limited clinical trial is just a proof of concept. But you really need to do a lot more to get to that particular stage where this can be done. The other questions, I think, these are the two interesting questions that I thought that were there. The other
one generic maybe, for somebody who is doing a bachelor's or master's and they would like how to join PhD in IISC Bangalore. And I'm currently doing MTech, so it's very generic, but
Yeah, so the question is very simply apply to IISc. So if you meet certain thresholds for MTech, these thresholds change a little bit from year to year, as you would imagine. Then if you are shortlisted, you are called for an interview. Now the interview session is pretty grueling. Let me put it that way, because it's not intimidating, it's grueling. So what we look over there is your fundamental understanding of stuff. So whether you remember the definition of Teckley number is not important. At least it's not important to me. Okay. What is more important is that whether you have the aptitude or the thinking process to basically, divide a problem into smaller blocks, how you approach the problem. For example, if you can understand what forces are acting on it, can you you may not know the non-dimensional numbers pertaining to this. That is still okay. But if you do that, the chances are very high that you would perhaps get in. But it's a tough competition because we normally would look for people with very high scores in the interview. So it's of course still there is a weightage. There is a weightage on interview. There is a weightage on your career so far. So fundamental understanding is of essence. If I have to put it.
And is there any way for international students to join a PhD at IISc?
Yeah, that's not a problem. They are welcome. They have to apply through the Office of International. This is called IRO, I think, International Relations Office or something like that. It has got an acronym. You can look it up on the website, and there it is obvious that you can actually, apply through that channel. You again have to go through the same process, except that there is a shortlisting criteria and then there is an interview. So everything happens through interview, whether you like it or not. That's how things work so far. So basically, we don't trust the testing agencies, if I have to put it that way. And I think for right reasons.
Yes. So another interesting question, maybe for early career researchers, like how to set up or what is the strategies to set up a lab, experimental facility. So any insight on that? Yeah.
If you want to, the context is very different if you are, because I have been a faculty in both continents, basically. So India is a lot different from the US. for example. I am not familiar with the European system. It may be a little different than that. So, or maybe a linear superposition of the two. It could be like that as well. So coming to the Indian context is that if somebody wants to join an Indian institute, like an IITs or IISc as a matter of fact, so the idea is that here the startup grants are quite reasonable these days. It's actually pretty liberal. So you have to know what you are planning to do. So it is better first to have a research proposal or basically a wish list of what you want to do, have some short-term goals and have some long-term goals. Short-term goals are something that you would need to get going because like cricket, research is also a confidence building game. So if you are inactive, if things are not working for a very long time, people tend to get their morale gets down and that affects your thought process. So I would say that have a balance kind of a thing, that have some short-term goals, which you can do immediately. Problems are something that you know that you can do, and maybe have some risky type problems, the problem or territories that you are trying to venture into where you do not know what is going to be the outcome, and have a balance between these two so that whenever you're working in the in the lab, or if you're a computational guy or an experimental guy, doesn't matter, you can get some quick bucks basically, which are good results. I'm not talking about the cut corners in your research, but something that you are very used to doing. So you can do that. And then, at the same time, build up slightly riskier projects, I should say, and execute them as and when you go. In that way, you will be always productive and you will be always appreciated as well. And at the same time, you will have a balance of both worlds. So this is the Indian scenario. The tenure is not a big problem in India. Okay, I mean, the tenure exists in IISc, but it's not something that I would be bothered about. Well, tenure, honestly speaking, doesn't really matter. You have to be content with yourself, with what you are doing. So that's the whole point. Whether it is linked with tenure or not is immaterial, according to me. So in India, of course, the funding situation is pretty good. So if you are sitting in IISc or IITs, the funding situation is pretty good. You can write proposals, you can get substantial amount of money in not a long period. These days, the turnaround time of the agencies are also quite good. So you can get a lot of, and there are a lot of schemes that are floating around. So getting a couple of crores should not be a problem really, apart from your startup. So that should get you going, and you can produce some things. And then you like in the US, you then try to go for bigger sized funding. But that will be only given to you when you have a credibility. Like, for example, the defense or ISRO, they are going to give you that kind of money when they're comfortable with you, that you are able to do what you are kind of in. So, and in India, well, to be very frank, sometimes things can go slow at the institute level. Whoever have done their PhD in India would know this for sure. So, and there can be a little frustrating at times. But the entire idea is that you have to kind of maintain your, cool, and you have to go on pushing the envelope, and it will get pushed. That much I can say. So, so there is no shortcut. It has to be just the amount of time that you spend and how you do it and stuff like that.
Okay. And one more thing connected to that, like getting funding from the DST or the agencies is one, and another is collaboration with the industry. Like for example, in Europe or USA, it is all the universities are, there is quite a lot of collaboration. So how is it progressing now from
India,
IIS?
it's picking up. Actually, the industrial part of it is picking up. In IISc, IIT Madras, and all these places, the industrial presence is slowly and steadily growing. And this I can say from my own experience that there has been an upsurge in the formation of startups, engagement of the existing industries into this. Even today, we had a meeting with St. Gobind, for example. So these things, previously what used to be not so good, I should say, has picked up. So the startup culture has picked up quite a bit, and that is an excellent sign. At the same time, existing industries are also somewhat, they are more receptive. I won't say they are very receptive, but they are somewhat receptive. Not like the Europeans still. We are not at that level, but we are going there. So this is, I think, a very good sign. So ideally, this plug-in with the industry actually serves two purposes. The PhDs need not always become faculties, because that is not the way the European the European countries work as well. I mean,
Yeah.
so faculties, I think their ratios are also very low because very few people go there. So, but their industrial presence, which requires PhD, is quite high. So I think we would rather go for that. So India should strive to build that ecosystem, and it's slowly happening. I mean, if you are in IIT Madras, I suppose, so you have seen the incubation park and stuff like that. And with guys like Satya Chakravarti who's doing Agnikul and all things, these are great. I mean, he was also a fundamental guy as far as I know. So he's doing great. So I'm also advising quite a few startups, development of microturbines and stuff like that. So we are, the scenario is also making us to push ourselves, which I think is a good thing because, professors normally are pretty, we have very high inertia. So the fact that we are also getting moved shows that the country is perhaps going towards the right direction as far as the industrial ecosystem is concerned.
That's great to know, sir. And one question is about, again, connected, like what are the future research directions like in next 10 years in which, in which area students should focus on? Maybe some insight in fluid dynamics related area.
So traditional areas are slowly fading quite a bit. So you have to have, for example, this data science bandwagon. Okay, you might criticize it in your drawing room that it is very bad. Okay, and actually, we are not very receptive to changes to begin with, but it is here to stay. And there are some very good points. Okay, so I would say, so data science driven. So I am not asking people to sacrifice their fundamentals. It is still the fundamentals, but you some way connect it. Be receptive to the data science like you would be receptive to any other things. Okay, so that is the way. I mean, so that is one thing. Then hypersonics obviously is a big ticket item, which I feel is going additive manufacturing. So at all scales, I'm not talking just fundamentals, from the manufacturing side to everything. So I mean, 3D printing, obviously, it's additive manufacturing and 3D printing. Biomedical, because this is the century of the biologists. You just look at the funding situation. A biologist will be raking in millions while we have to break our head for a few dollars. So essentially, it's the age of the biologist, and for the right reasons, because we have know a lot about biology. So it's instead of kind of shying away from biology, it is better to embark on that biology and try to see how we can contribute. I'm not asking people to become biology, but as I showed in some of the applications, we can link that to. And there are people who develop drug, delivery using the fluid mechanics background or physics background. Okay, so we can actually join hands with the biologists, and that can be a great area of research, according to me. And of course, ideas like this lab on a chip type of, which is once again biology, so to say. I mean, traditional may not be. I mean, traditional things, but microfluidics is slowly fading away. So you need interjection of new things into that particular paradigm. So that's why we say something like lab on a drop and stuff like that. So you have to invent, you have to push all these frontiers a little bit beyond. If you just stay in your comfort zone and do what the old school fluid dynamics people did or combustion people did, then it's not going to work because the field has progressed. And I would say that it should progress. I mean, why would we remain in that particular limbo?
Exactly. So collaborating with different community and different fields.
Absolutely. You learn a lot from each one of them. Okay, I have collaborated with the material scientist, for example. So there we could contribute. Okay, in his area, which is his core domain, biomaterials. But we could contribute. And the contribution can come through different ways. It can come through fluid mechanics. Somebody who's doing solid mechanics is also a part of it. So you can actually go on, doing a lot of stuff. More interdisciplinary, more multidisciplinary teams. US, you have the concept of Moody, for example, multi-university research initiative. Something like that is required even in India, where you basically have this center of excellence, which combines a lot of people. Essentially like a Manhattan Project, but you are like, in a scale of the problem that you are concerned with. So that should go a long way in basically, solving or pushing the frontier, and you also will remain relevant in the field.
That's exactly. So, and one more about the liquid hydrogen or hydrogen combustion, because that's another field where a lot of,
Yeah,
like I can
yeah.
see in Europe, there is a lot of funding happening.
India
So,
too. India too. India too.
okay.
It's the same thing even in India. And it's all this green, blue,
Yes.
green. What are the gradations? I just kind
Yes.
of
Green.
get
Yes.
it. So all these hydrogens and then decarbonization. That is also a big
Yes.
area of research. I mean, so again, the application is hydrogen and decarbonization, but the methods that you use has got a lot of fundamental stuff, and then it has got a lot
Yes.
of application stuff. So you have to
Exactly.
get to these things, and these are relevant problems because you have to save the planet. Okay, you have to decarbonize everything because we have done a lot of damage to the planet. So the planet needs to heal and all those stuff. So there is a greater good, but at the same time, you don't leave your field. You can still do
Yes.
combustion. Do it with hydrogen combustion, and there's a lot of things that you can do with hydrogen combustion.
Yeah. Because that was one of the question or a concern, like, now EV is coming and where is combustion going? So, this
Yeah.
is another field, like
Well,
we
it's
just
going
need
to
to change.
stay. Well, it's going to
Yes.
stay because the problem with EV is that you basically localize the pollution or the
Yes.
toxicity to local pockets.
Yes.
So all you have done is that you have taken a uniform flow and you have made it like kind of sit in kind of
Yes.
all the peaks.
Okay.
So it's not a good idea. So going to something like green combustion, hydrogen being one of them, and then hydrogen production is an issue that one has to address. So those kind of so combustion is not going away. Definitely, it is not going away. Okay, it will just reinvent itself in a form. So see, all these
Yeah.
waves comes and all this goes. Fuel cell was a wave at one point of time. It served its purpose. So I think similarly, EV, while it serves some purpose, it keeps you locally clean. But I think in combustion is a much more proven technology than that.
Yes.
Okay, because you burn, you get something. I mean, this is a
Yes.
very basic law which has been there when mankind first came into existence, probably.
Yeah. So I think we have exceeded 20 minutes, but sir, thank you very much for
Thank
your
you.
time and coming for our invitation. So once again, thank you, sir. And I
Thank you.
think
If I can have, if I think you have recorded it, if you can send me
yes,
the
yes,
recording
yes, I will share you the recording and also will upload it on the YouTube and then share that as well. Once
Great.
again,
Thank you. Thank you
thank you
so
very
much.
much, sir.
Thank you.
Thank you,
Thank you so much for the invitation and bye.
thank you. Bye.
I'm going to leave.